So how do humans go from a single cell to a complex multicellular organism? That's what we'll be talking about in this video. Embryonic development begins with the fertilization or union of sperm and egg to form a zygote. Copulation or sexual intercourse releases hundreds of millions of sperm into the vagina, but of those, only a few hundred survive the trip to the egg, and of those few hundred, only one will fertilize it. A mature human sperm cell has a streamlined shape that suits its need to swim through fluids in the vagina, uterus, and oviduct. That sperm cell needs to pass both a layer of follicle cells to get to a barrier known as the zona pellucida. The sperm cell then releases enzymes which digest through the zona pellucida, allowing it to access the plasma membrane of the egg cell. The first sperm cell to do this fuses with that egg cell, releasing its nuclei into the egg to fuse with the nuclei already present in the egg. The result will be a diploid zygote nucleus, containing chromosomes both from the mother and the father. The early stages of human development begin with fertilization and cleavage in the oviduct. At some point after ovulation, if sperm and egg meet, that fertilization occurs within the oviduct. There's a series of rapid cell divisions known as cleavage, and as that developing egg continues to travel down the oviduct towards the uterus. So development begins with cleavage. A series of rapid cell divisions that results in a multicellular ball. In humans, cleavage continues as the embryo moves down the oviduct towards the uterus. About 6 to 7 days after fertilization, the embryo has reached the uterus as a fluid-filled hollow ball of about 100 cells called a blastocyst. About 1 week after fertilization, the embryo, which has become a blastocyst, implants itself into the uterine wall. The outer layer of the cells of the embryo, the trophoblast, become part of the placenta. So we see before implantation, there's a trophoblast with an inner cell mass, and as this then reaches the uterine lining and embeds itself into the endometrium, the trophoblast cells divide, becoming the placenta, and that inner cell mass continues to divide to become the developing embryo. Pregnancy or gestation is the carrying of developing young within the female reproductive tract. It is measured as 40 weeks from the start of the last menstrual cycle in humans. Now, it's important to note that this 40 weeks, it isn't measured from the time of intercourse. It isn't measured from the time of implantation. We don't always have that information. It's measured as 40 weeks from the start of the last menstrual cycle. Pregnancy itself is divided into 3 trimesters. Here we see a photo of a human embryo in the first trimester. By the end of the first trimester, the fetus looks like a miniature human being. The anatomical sex of the fetus can be determined by ultrasound. Now, I asked my wife if it would be okay to share these pictures, and she said it was. These are the first pictures of our oldest son. His picture at 9 weeks and his picture at 18 weeks. Now, these pictures are not my son. These come from the textbook. The main developmental changes during the second and third trimesters involve an increase in size and general refinement of the human features. We can see here at week 20, eyebrows and fingernails are already present. At birth, a typical baby is about 50 centimeters, or 20 inches in length, and weighs 2.7 to 4.5 kilograms, or 6 to 10 pounds. And this is a picture of our oldest son, Perrin. So what happens when the genetic instructions aren't exactly correct? That's what we'll be talking about in the next video.